Welcome back to Dr. Liu's VCE Biology. Today's topic is the relationship between nucleic acids and proteins, as presented in the 2018 VCE Biology exam. Let's learn together. If teachers, parents, or students notice mistakes in my explanations, please provide me with your feedback. While the genome is a stable blueprint, the proteome is the dynamic, ever-changing set of all proteins in a cell, tissue, or organism. The study of this entire protein collection, a word coined using the -ome suffix to denote the whole, or total set, is known as proteomics. It reveals how proteins function, interact, and contribute to biological processes and diseases. Two adjacent amino acids combine to form a peptide bond in a condensation reaction that eliminates a water molecule. Unlike many such reactions, however, this process is endergonic, meaning it requires an input of energy. This energy is provided by ATP hydrolysis, and the reaction is catalyzed by enzymes. While the peptide bond is thermodynamically unstable, its high activation energy makes it kinetically stable, preventing it from breaking down without the help of enzymes. The genetic code is degenerate, meaning multiple codons can specify the same amino acid. This term, while originating from the Latin degenerare, to fall from one's kind, is used neutrally in science to describe a many-to-one relationship. This redundancy is a crucial protective mechanism against mutations, as a single change in a nucleotide may still result in the correct amino acid, which helps prevent a change to the final protein's function. The diagram below represents an aspect of the activity of the Lac Operon, a group of genes found on the chromosome of the bacteria Escherichia coli. The diagram shows the events that occur if allolactose, a breakdown product of lactose, is present in the E. coli cell. Enzymes like transacetylase, permase, and galactosidase are proteins, and the instructions for their creation are found in exons, the coding segments of a DNA sequence. A separate DNA sequence, the operator, acts as a binding site for a repressor protein, molecule Q. This repressor is produced by a regulatory gene, and its binding to the operator prevents the expression of the structural genes. When the repressor protein, molecule Q, binds to the operator, section X, it physically blocks the RNA polymerase from moving along the DNA. This action prevents the transcription of the genes that are located downstream, further along the DNA strand, effectively turning off their expression. This mechanism is a fundamental way that cells control gene activity, ensuring that proteins are only produced when and where they are needed. A single amino acid change, like proline mutating to alanine without any other amino acids being lost or gained, is the most likely result of a nucleotide substitution. This type of point mutation involves a single nucleotide being swapped for another, which alters just one codon. The mutation is not a deletion or insertion, as those would cause a frameshift that changes every amino acid from that point on. Similarly, it is not an inversion, which would also significantly disrupt the entire peptide sequence. A mutation that results in a shortened, or truncated, protein is most likely a nonsense mutation. In this specific case, the codon for the amino acid tyrosine, located immediately after valine, mutated into a stop codon. This caused the translation of the mRNA to stop prematurely, producing the shortened protein. In humans, Duchenne muscular dystrophy, DMD, is caused by mutations in the dystrophin-encoding DMD gene. The DMD gene contains 79 exons. If a patient has a duplication of one exon, the translated protein will be longer than normal only if the number of nucleotides in that duplicated exon is divisible by 3. This is because the genetic code is read in triplets, codons, and a duplication, that is a multiple of 3, adds a full set of codons without causing a disruptive frameshift mutation. The synthesis of the enzyme tryptase is directed by instructions encoded in nucleic acids within a mast cell. The process begins when a ribosome binds to the mRNA messenger RNA transcript. Transfer RNA, tRNA, molecules, each carrying a specific amino acid, are brought to the ribosome, with the tRNA anticodon being complementary to the mRNA codon. The ribosome then joins these amino acids together via condensation polymerization to form the tryptase protein. This protein, along with other chemicals like histamine, is then released from the mast cell. After being synthesized in the rough endoplasmic reticulum, RER, the enzyme tryptase is transported to the Golgi apparatus. In the Golgi, the protein is modified, sorted, and packaged into vesicles. These vesicles then move to the plasma membrane, where they fuse and release their tryptase from the cell via exocytosis. The energy required for this entire process is provided by the cell's mitochondria. This section is worth 14 marks and contributes to 11.7% the total 120 marks. Multiple choice questions, 8 marks, Q2, proteome, the full, dynamic set of proteins in an organism. Q3, condensation, amino acids are joined to form a peptide bond. Q4, degeneracy, multiple codons specify one amino acid, protecting against mutations. Q5-6, Lac Operon, a repressor protein binds to an operator to regulate gene expression. Q 2021.0. Nonsense mutation, a single nucleotide change that can create a premature stop codon. Q 22. Exon duplication, duplicating an exon with a nucleotide count divisible by 3 adds amino acids without causing a frameshift. Short answer questions, 6 marks, Q1a, protein synthesis, the process of transcription, DNA to mRNA, and translation, mRNA to protein. Q1b, protein secretion, the journey of a protein from the rough ER to the Golgi apparatus, and its release via exocytosis, powered by mitochondria. If you need my help, please add me on WeChat or WhatsApp, and send me an email. Thanks for your attention. Please note Dr. Liu's VCE Physics channel, Dr. Liu's VCE Chemistry channel, and Dr. Liu's VCE Biology channel.